Dr. Teng, are you there and ready?
I am here.
All right, great. I think we'll get started in just 1 minute.
That's good.
Teng, how are you, sir?
Good. How are you?
I'm great. Dr. Lazama sends his regrets. He's up in Boston for this ceremony.
Yeah.
I understand he's having a good time. They're having some good speakers, but you get me today as your introducer.
That's great.
Hey, Mike, Dick Lockey, thanks.
Hi, Dick. How are you?
Dr. Lockey, how are you, sir?
Hi, Faye, how are you doing?
Doing great, thank you. All right, good noon, everyone. Jen, is it okay if we get started? I'd like to be on time.
Yes, all set.
All right, welcome everyone to Grand Rounds. My pleasure to introduce Dr. Michael Teng. I was looking over your CV, very impressive. This is, to me, sort of the classical academician's CV. Started his studies at MIT, Bachelor of Science in Life Sciences, and then moved to Chicago to do his PhD. Numerous stops along the way from California to Pennsylvania before arriving in Florida, 2010, in the Department of Internal Medicine, where he has, and throughout his stops, I was noticing that Dr. Teng has dedicated himself to teaching, to research, to course development. So he's got a plethora of accolades and experiences on the teaching front. So truly an academic's life he has led. Currently, very impressively, including patents, by the way, a number of patents, and we're not going to go over all the details. It would take into your speaking time. But currently he's got an R01 that just started a year ago, about 4 or 5 more years left on the R01. So hopefully we'll hear a little bit about your research endeavors. I'm trying to think if I'm missing any of the majors. I think that's the key highlights. I don't know you personally to talk about your hobbies and your interests. So you're welcome to fill in the blanks, but we are certainly grateful to have you on faculty, grateful to have you here today, and look forward to your talk.
Yeah, thank you for that very nice introduction. Don't have a lot of hobbies because I got a little girl that needs to be driven around a lot, but
oh, you and I have the same hobbies. We are chauffeurs.
Yes, we are the Ubers for our children. I
yes
would like to point out one of the things that Dr. Yusuf mentioned is that one of those patents that I'm on was actually a collaboration among a number of different groups here, both in the Department of Internal Medicine, but also within the College of Medicine. So it's been a really fruitful
Was this
research.
Summer Decker's the swabs? Yeah.
Yep, Summer Decker's swabs. That was done in collaboration with Dr. Kim's division of infectious diseases. So this was a fun project, and I think a pretty useful one early in the pandemic.
Yeah, so those of you who are new to us, during the pandemic, there was a shortage. I'm not going to do the story justice, but a shortage of swabs. And Dr. Teng, you and Dr. Decker and that team developed a 3D printing model for it and won some prestigious awards as a contribution to the pandemic. But the stories are pretty colorful because there was some political interest in this scientific achievement to the point where there was some pressure to, take ownership of it by the state, other different, financial interests, and the group, to their credit, said, no, this is for humanity. We're going to put the formula out there. We're going to put the program out there. Whoever needs it gets it. And you've probably on some of these letterheads and calls from various countries around the world thanking you for this contribution. So this is a major achievement from the faculty at USF. So congratulations and thank you 2 years later for that contribution to science.
Yeah, thanks. It was a really fun project, and I really enjoyed working with our colleagues both in medicine
Were
as
you
well as
sleeping
radiology.
in the office
No, that
24/7 too? No
was
showers?
Dr. Decker and Dr. Ford. What my job for them was to bring them food and make sure that they were properly, hydrated and fed so that they could get a lot of the 3D printing done. And then we coordinated a lot of our med students actually helped package the swabs and make test kits and stuff like that. So it was pretty much an all hands on deck effort. It was great.
We'll wait for the Hallmark TV version of it to come out.
Right.
It's a good story. It's a really good story. So without further ado, welcome and thank you for being here.
Yeah, thanks. Thanks again for the introduction. So today I thought I'd talk a little bit about vaccines. We've heard a lot about vaccines over the course of this COVID pandemic. I think one of the things I wanted to do here, let's see, a laser pointer, was really just go over some of the background on the vaccines. We've had, a couple hundred years of vaccination since Edward Jenner started using cowpox for vaccinating smallpox. But over the course of time, and especially really now in the last couple decades, we've developed a lot of different types of vaccines. So I thought it'd be interesting and kind of informative for me to go over the different types of vaccines, what they're good for, how we make them, what they're not good for. And I would talk about the new vaccines that have come out recently, some of the newer technologies, and how this has improved our vaccination procedures. And then I wanted to go over the CDC vaccination recommendations. These are they're now that we have more and more vaccines, they tend to get more and more confusing what exactly we are looking at in terms of what vaccines are recommended at what age and which categories. And then I like to finish off by just highlighting a few of the vaccines that are out there that have specific recommendations or have specific interesting characteristics. Okay, so just to begin, kind of in a most general sense, why do we vaccinate? There's both the micro and the macro part of why we vaccinate. First off, obviously, the micro is protection of the individual. We want to make sure that we don't get infected again or that we don't suffer the severe consequences of those infections. But really, the secondary benefit for these vaccinations is to protect the community, particularly when we can block the transmission or reduce the transmission of viruses, of pathogens that can help the people who can't get vaccinated in order to protect them from disease. In very rare cases, we have actually been able to cause the extinction of one virus so far, smallpox, and potentially another, polio soon. So this is another possibility that we can use if we have mass vaccination programs that are really coordinated and effective. This is just kind of to give you an idea of how well this public health effort with the vaccinations works. These are older vaccines, so diphtheria, tetanus, MMR, pertussis, things like that. So these were all available before 1980, and so they've been deployed in our arsenal of vaccines for decades now. And you can see that before vaccinations occurred, and look at some peak years for diphtheria in the 30s, 30,000 cases, 3,000 deaths, basically zero in 2006, 2004, and 2006 when this JAMA article was published. So you can see we've actually had pretty good extinction of these diseases, or we don't see these diseases very often anymore, at least in the United States. So diphtheria, for example, polio, poliomyelitis, smallpox, even rubella. Very, very infrequently we see rubella. Measles virus cases now tend to be mostly imported from other countries that have not controlled measles virus. So we're seeing, virtually 100% reduction in deaths and cases in a lot of these for a lot of these diseases for these older vaccines. Newer vaccines are similarly effective. We haven't gotten to the point where we have enough coverage or we have enough kind of impetus to try to vaccinate everybody. But if you can see that we have some very good vaccines for hepatitis A and B, Hib, pneumococcal disease, varicella. These, again, also have very good decreases of hospitalizations and deaths from these diseases now with these available vaccines. So this is really important both from the protection of the individual, but also the spread of these diseases. Okay, so we have these vaccines. The idea of a vaccine is, this is going back to basic immunology. It's basically you're priming your immune system to recognize a pathogen once you get infected again. This is really the generation of immunologic memory for rapid secondary response. And this secondary response comes up obviously more rapidly and to a greater extent so that you get rapid elimination of the pathogen once you come into contact with it again. This is the basic immunology version of vaccination. So the idea really is that your primary response is very low and it comes up later because your immune system has to learn to recognize the pathogen. But once it has already learned how to do this, the second time you see that same antigen, you get a really rapid rise of your adaptive immunity that's recognizing specifically that pathogen. And you can then see the control of that infection during this secondary response. So there are a lot of factors that go into this, getting a proper secondary response, having to do with genetic factors, how the immune system is working in the particular vaccinee, how vaccines are actually given and how those vaccines can induce immunity. Some are more effective than others at inducing full immunity, but these are differences that we'll get into when I explain the different types of vaccines that we have. But really, the whole idea is do this, vaccinate in a way that is safe and teaches your immune system so that when you do have to have the immune response with the pathogen exposure, then you can actually get really rapid responses and clearance of the pathogen. Okay, so we didn't really appreciate probably before 1980 how these immune responses worked necessarily. I think a lot of the discussion, even back like 30 years ago, when we talked about immune responses, a lot of the response to vaccinations were really focused on the humoral response. This is the development of your antibodies, your B cells, plasmablasts, in order to have this kind of surveillance in your serum so that you have circulating antibodies to protect you from infection. I think more recently, in the last 30 years, it's been pretty clear that the T cell memory response is also very important to make sure that is that's recognized as well. So more of the vaccines now are really trying to incorporate also the cell-mediated response in addition to just the antibody response. And pretty much the newest thing for vaccinologists is really to make sure that you're getting proper mucosal immunity. Mucosal immunity is one of those things that's really difficult to study because we don't have really great ways of looking for markers of mucosal immunity. If you're looking at a respiratory infection, getting samples from the lungs or trying to, determine immunoglobulin levels in the lung parenchyma is very difficult. But the idea with mucosal immunity is that if you have these resident memory cells in the mucosa, that you get a quicker response, and that response actually is able to work better in that particular region, in that particular tissue, rather than having to generate a systemic response that then migrates to the tissue of interest. Okay, so there are a lot of things that we have to think about when we're making a vaccine. I think, in terms of just the generation of the vaccine, you want to make sure that you get a protective immune response, but that protective immune response has to be the appropriate immune response. And what we found over the course of many trials and errors in the vaccine field is that you do have to have kind of a balance of cellular and humoral immunity before you can get a really good immune response that will clear the pathogen once it comes into your body. You do want to have kind of a broad-based immune response so that you don't have to change the vaccine or have multiple vaccinations. So immunity against a variety of strains is also important for a good vaccine. Some of the best vaccines that we have give long-term protection so that you don't have to give multiple doses over the course of your life. Obviously, the thing that we test for in clinical trials is safety. Safety is always the first consideration for phase 1 and phase 2 trials. And this is why a lot of our vaccines initially have been kind of dead vaccines, vaccines that don't replicate. Ease of administration, stability, and cost effectiveness really are those considerations for vaccines to allow for them to be deployed in different public health settings, not only in our large urban academic medical centers, but in rural areas of our country, both here, but also if you think about these diseases, most of them are global diseases. So how can we get these vaccines out also to people in low resource countries where you might not have freezers or refrigerators, that travel is more difficult. And especially low middle income countries cannot afford to buy large amounts of very expensive vaccines. So the idea of getting an easily deployable vaccine is also very important. Okay, so what I'm going to do next is really just kind of go through the list of different technologies that we use to make vaccines, starting with live attenuated vaccines. Starting with the live attenuated vaccines because the cowpox really was the first vaccine. Vacca for cow in Latin is where vaccines actually come from. And so the idea of a live attenuated vaccine is somehow you make that pathogen that you're trying to vaccinate against weaker so it doesn't cause disease. You can do this by attenuating the virus through passage in a foreign host, so basically changing the host range of that pathogen. That's been done for a lot of the vaccines, live attenuated vaccines that we currently use. Jenner's cowpox virus was using a heterologous virus, so a cowpox instead of smallpox. You can still, in this case, get an immune response that recognizes smallpox if you vaccinate with a different species of orthopox virus. These have been really effective. The original attenuation through passage in a foreign host was something that was done with rabies virus for Louis Pasteur. So these are technologies that are a couple hundred years old at this point. The newer types of technologies, though, are trying to do a little bit more rational design of live attenuated vaccines. So basically, the idea is to learn about the virus or learn about the pathogen, try to take away those virulence factors or those proteins that are really essential for the pathogen to replicate well in the host, and reverse engineer the pathogen so that is now attenuated, and you've done it in a very rational way because you've taken out specific proteins that will weaken the pathogen but will still allow for the antigens recognized by your immune system to be expressed to high levels. So we have lots of good things with these live attenuated vaccines. These live attenuated vaccines produce a really broad-based immune response because you're immunizing against pretty much the whole virus or bacterium. Lots of different proteins. You have both cellular and humoral responses because that's what your body does normally to get rid of the pathogen. These are replicating viruses, or replicating vaccines, so they can actually make a longer lived and a more broad response in the immune response, and that also means that most of these live attenuated vaccines are a single dose. Because they are replicating vaccines, they're inexpensive because you don't have to inoculate with very much of the vaccine, so they're easier to make. The problems, of course, are that these are still replicating vaccines. So you have the potential for these live attenuated vaccines to revert to virulence, and you also have the problem that these live attenuated vaccines, for the most part, can't really be used in immunocompromised people because they don't have as much control over, they won't have as much control over the infection. The other thing, just in terms of public health, is that these live attenuated vaccines generally are very sensitive to storage conditions. So you need a cold chain, you need freezers, you need some way of keeping the vaccine stable enough so that it can be deployed into resource-poor areas. We have a lot of live attenuated vaccines. And what I'll show you really is that a lot of these vaccines that are live attenuated tend to be pediatric vaccines. So maybe not something that you're going to see in your practice as often in a medicine department. But these are some of the best vaccines that we've had. Measles virus vaccine is one of the best vaccines that we've had besides the smallpox vaccine. So MMR, the pediatric version of the chickenpox vaccine, varicella zoster, something that's not used in this country anymore, the oral poliovirus vaccine, rotavirus vaccine. Some of the other ones are more are only for adults, really. So there's a live attenuated influenza vaccine, yellow fever, which is given mostly to travelers into yellow fever endemic regions, Mpox, which is the same vaccine that we used for smallpox, which we still had something like 30,000 cases of Mpox in the last couple of years. There are two new live attenuated vaccines within the last 2 or 3 years, one for cholera and one for typhoid. So these are also live attenuated vaccines. The oral typhoid vaccine is live attenuated, and this also then goes to the idea of mucosal immunology, where you're actually taking it orally and getting a good immune response in the GI tract. Okay, the second type of vaccine that we started using were inactivated vaccines. So the problem with some of the live attenuated vaccines are if you don't attenuate the vaccine enough, you can get disease. So then the next logical idea was to take the virus itself and just kill it. So you take the virus, you kill it, and then it goes into it still has the antigens and goes into the it induces the immune system, immune reactions to that virus. These are also there are also inactivated vaccines for bacteria as well, but most of the inactivated vaccines are viral vaccines. So what you do is to produce large quantities of the virus pathogen, and you basically kill it with chemicals. And what we normally use is formaldehyde. It's incubated with the pathogen for a number of days. You wash out the formalin, and then that alone is the vaccine antigen that you're using. There are some other chemicals that have been used. Formaldehyde obviously has some negative effects. So people have been trying other chemicals. Beta-propionolactone can be used as well. It's not as common because it's a little more difficult to make, but beta-propionolactone has the benefit of being able to be hydrolyzed to non-toxic substance. The big problem with these inactivated vaccines, though, is that when you chemically destroy the infectivity, especially with formaldehyde, formaldehyde works by cross-linking lysines. So if you cross-link these proteins together, you may disrupt some of the immunogenic epitopes and not be able to get that full neutralizing antibody response. But we've used a lot of inactivated vaccines, and these do have a number of advantages over the live attenuated vaccines. First off, that they're non-replicating, so you're not really going to get disease or disease from the pathogen. You may get local reactions at the site of injection. If you have adjuvants or anything else in the vaccine, you may also get reactions to that. But basically, there should be no major systemic effects from the vaccine itself. Also, inactivated vaccines are very stable because they are inactivated. They are basically killed. You can lyophilize them, put them into a vial and leave them lyophilized so that you can reconstitute it at the point of care. The big problem with these inactivated vaccines is because it's a non-replicating vaccine, and you've basically all of the antigen that you're going to give is given in that initial dose. So a lot of times what you see with the inactivated vaccines, you only see B cell responses, you only see antibody production. You often have to give multiple doses of the inactivated vaccine over the course of life in order to maintain protection from disease. And there are some cases, both measles virus and the virus that I work on, respiratory syncytial virus, that the inactivated vaccines can cause an imbalance in the antibody response and result in atypical disease. It's this really big problem with this atypical disease that makes vaccine enhanced disease part of every clinical trial. So there's always a flag for vaccine enhanced disease because of this imbalance in antibody production, both for inactivated vaccines as well as some of the live attenuated vaccines as well. One of the big problems as well for the inactivated vaccines, because they give you a poor response, is that you need to add adjuvants. Adjuvants tend to be different molecules that will stimulate your immune response in order to enhance the immune response to the antigens that you're giving. For a long time now, we've been using just a few different types of adjuvants. Most of the adjuvants that we use are aluminum salts. Aluminum salts really have a number of probably different mechanisms of being an adjuvant, but one of the reasons that they're thought to be effective is that they kind of deposit all of the antigen into your antigen presenting cells, your dendritic cells, in order to stimulate a better response. These are safe, they're effective, but they tend to skew the response towards humoral immunity. So more recently, people have been trying to figure out different adjuvants to get a better T cell response in addition to the B cell response. People have used liposomes. We'll talk a little bit about the lipid nanoparticles that are used in the mRNA vaccines for COVID. But a lot of the adjuvants that are available with these inactivated vaccines are things like pathogen-associated molecular patterns. So these are things that stimulate your innate immune response, stimulating toll-like receptors, stimulating Rig-like receptors. And one of the kind of most widely used PAMPs that's in an adjuvant is monophospholipid A, so the backbone to LPS or endotoxin that stimulates immunity through toll-like receptor 4. In addition, there are chemicals called saponins that are derived from tree bark that are also very effective adjuvants. They can stimulate mainly T cell responses as well. And then there are combinations of some of these adjuvants, and the kind of classic combination that GSK uses is this AS01, which is a combination of monophospholipid A and saponins. So we have a whole bunch of inactivated vaccines that we still use. The flu vaccine, for the most part, we use are inactivated influenza viruses. So you grow up a lot of the influenza virus, formalin inactivate it and inject it into intramuscularly. Hepatitis A is inactivated. Poliovirus, the inactivated poliovirus vaccine that we do use here in the United States now is inactivated. Rabies virus, which can be given post-exposure, one of the few vaccines that works post-exposure because you're trying to protect against the neurologic symptoms of rabies. That is an inactivated vaccine as well. And then a very kind of uncommon vaccine, Japanese encephalitis virus, is inactivated as well. More recently, though, we've been trying to develop better vaccines and using the idea that you're going to use maybe a viral vector, something that can replicate a little bit or not too much, but can deliver just the vaccine antigen to the proper immune cells in order to develop that balanced humoral and cellular immunity. So people have been using replication incompetent or heterologous viral vectors in order to deliver antigens. The one drawback of this is that you're making a single antigen as the potential, the antigen for vaccination. There are a few different types of viral vectors that are used. Adenoviral vectors, which we're familiar with the COVID vaccines. Vesicular stomatitis virus is used, which is a livestock pathogen, does not cause much disease in immunocompetent adults. A derivative of the original vaccinia virus called modified vaccinia ankara, MVA. This is used to deliver vaccine antigens. And the yellow fever vaccine itself has been used more recently as a heterologous viral vector in order to deliver vaccines. And that yellow fever vaccine virus, because it's such an effective vaccine, has been used for dengue virus, Dengvaxia, which is approved in the United States. SARS-CoV-2, obviously the adenoviral vector from J&J and AstraZeneca. And then there's a vaccine for Zaire Ebola virus. There are two. The one that's approved in the United States is for vesicular stomatitis virus. The EMA has approved an adenovirus MVA combination vaccine. So these are actually deployed. They're relatively new vaccines within the last decade or so, as our technology has gotten better in developing viral vectors that can replicate but also not cause disease. So we also have what we call subunit or recombinant vaccines. These are also some of the older vaccines that we have. So subunit vaccines really were developed by trying to take bacteria, break them apart, and just isolating those proteins that are immunogenic that will allow for protective response. So for tetanus and diphtheria, we use the toxins themselves. For tetanus and diphtheria, they're detoxified, and those are isolated from the bacteria itself. Pertussis is called acellular. It's still a subunit because you grow up pertussis, the pertussis bacterium, you break it open, and there's 3 different antigens that are isolated from pertussis. But when you're using these subunit vaccines, you're using proteins that are derived from whole cells that have been grown up in a production facility. More recently, with the advent of recombinant DNA technology, we've been able to make just the antigen itself without having to go through the process of trying to break open cells and trying to get away all the different components of the pathogen. And so you can make these proteins alone in your cell of choice, either bacteria or in yeast, in insect cells, and then isolate rapidly the protein itself to be used in the vaccine. So there is one influenza vaccine that is made from only protein, recombinant protein. The hepatitis B is probably the first of the recombinant vaccines, the hepatitis B surface antigen. The new-ish varicella zoster shingles vaccine, Shingrix, is a recombinant vaccine. But what I'd like to highlight really are these last 2 guys, because these guys they're relatively new, and they actually use a little bit newer technology than just making a recombinant protein. So this is the papillomavirus vaccine, which makes what's called a virus-like particle, and then the Novavax SARS-CoV-2 vaccine, which is a nanoparticle. So the papillomavirus vaccines, these first came out in 2006. And what it does, they take advantage of a very particular trait of all of these small non-envelope viruses. What these guys do is to make the outside protein, the capsid proteins, and these guys self-assemble. So what they found with the papillomaviruses is that there's basically 2 proteins, L1 and L2, that comprise the entire capsid of the protein, of the virus. That's all it is. And so those are obviously the immunogenic proteins. So what they did was just to express these proteins. If you normally do that in the context of a viral infection, if you have the viral genome, the genome gets incorporated into the viral capsid. These L1 and L2 proteins just come together. The L1 forms pentamers, which are then called capsomeres. These are the kind of Lego building blocks of the virus itself. And these capsomeres will then, 72 of these capsomeres will come together and form the viral capsid. These L2 monomers will then insert into the vertex of the capsomeres. And this is basically the authentic virus. This is what happens in viral infection. If you just express L1 alone, you will get something that looks like the virus. It just doesn't have the genome or the L2. And so this then is a great way of having the protein, because not only are all the kind of linear epitopes of the L1 expressed, but you get this kind of conformational expression of all of the different epitopes. So this actually is what your immune system sees, and it looks to your immune system like a real virus particle. And so this was a pretty neat way of using the virus's biology in order to make a really effective vaccine. So when these guys first came out, there was GSK and Merck came out with their papillomavirus vaccines about the same time in 2006. Merck's was a quadrivalent vaccine covering the two major cancer-associated papillomaviruses, types 16 and 18, as well as the two major genital warts-associated papillomaviruses, 6 and 11. GSK's only did 16 and 18. And I think GSK lost enough market share that they discontinued their vaccine. In the intervening years, because it's relatively easy to make these. If you can make recombinant protein, you can make different vaccines. Merck has now included 5 other strains of papillomavirus in their Gardasil formulation, so that's nonavalent now, 9 different types of vaccines, and this covers basically 90% of cervical carcinomas. I think it's something like 70% of oropharyngeal carcinomas in males, and then most of the genital warts causing papillomaviruses. So this is relatively easy. Now you can have a whole bunch of different antigens, and they all look like the kind of native virus particle. So this is a really cool idea. It's probably only good for those viruses that have this kind of outer shell that self-assembles, but it does give you a little bit of insight as to where this type of vaccine development is going. The other thing that is being done, so Novavax came up with their proprietary technology, and the idea here is basically the same idea as trying to make a virus particle, but trying to do it in a way that doesn't make a virus. It doesn't make a whole virus particle. This technology is really more useful for those viruses that have envelopes, that have a lipid bilayer, instead of just having the capsid protein on the outside. So viruses that have this lipid bilayer have proteins that stick out. So for SARS-CoV-2, that's the spike protein. What they can do is to make the spike protein in a cell. They use insect cells. They have a polysorbate 80 core. So this is kind of a hydrophobic core. And then they can stick these spike proteins into this core. And so when these are kind of spherical cores, little particles, and when you get a whole bunch of these spike proteins stuck into the particle, it kind of looks like the virus itself, because you have all of these spike proteins in the proper orientation, sticking out their heads, which are the kind of the business end of the spike protein. And so now you have that, you have an adjuvant, they have their own proprietary adjuvant, and this now, the nanoparticle itself kind of looks like a vaccine. You have an adjuvant to increase immunogenicity. So this is another way of trying to make a vaccine that's sort of multivalent and allows your immune system to get a better, more robust immune response. So Novavax is obviously using this for a number of different proteins as well, using this technology to stick in these proteins that would normally be in the envelope of viruses in order to form the nanoparticle vaccine. Okay, so last couple types of vaccines are polysaccharide vaccines or conjugate vaccines. Polysaccharides are, the kind of sugars that are on the outside of bacteria. You can immunize to that and make good antibody responses so that you can get rid of the bacteria. It turns out the polysaccharides alone are not really all that great at inducing immune responses, so they've actually started conjugating the polysaccharides to different proteins and use those conjugates as the immunization antigen. So we have lots of these guys, HIBs, so Haemophilus influenza, meningococcal strains, pneumococcal strains, and then typhoid. The typhoid vaccine that's approved is just a polysaccharide vaccine, while the other 3 here are conjugate vaccines. And the most recent type of vaccine that we've seen, really the most recent technology that we've seen, are these mRNA vaccines, which right now only have the SARS-CoV-2 vaccines that are approved, although with the success of those vaccines, there are a lot more in the pipeline. The mRNA vaccines work in a little different way. They actually introduce an mRNA into the cell and make the cells express the antigens. So now you can get expression of the antigen in a similar fashion as if you were being infected with a virus, but you don't have the whole replication complex of the virus. You only have the antigen itself. There are a couple different types of mRNA vaccines that are being tested. What is approved is these non-replicating mRNA vaccines, but the self-amplifying mRNA vaccines are kind of on the horizon as well. Just to show you what that really means, a non-replicating mRNA is basically encapsulated in some sort of lipid nanoparticle. It is taken up by the cell. You express the proteins in the cell, and those expressed cells then can be presented by your MHC molecules to T cells. It can actually be secreted or produced at the surface of the cell to be recognized by antibodies. So non-replicating mRNAs get in. Once they're there, they make the protein, they get degraded, they go away. Self-amplifying mRNAs are basically the same idea, except they include a replication protein, a polymerase from a virus, usually from a non-toxic virus. And this replicase will then replicate the RNA. So you can make more of that RNA in that cell. So you get more antigen for an extended period of time, because the mRNA doesn't just get degraded. It actually makes more of itself. So these guys all require some way of getting in the cell. There are a lot of different ways of getting in the cell. The most efficient way is this lipid nanoparticle that includes polyethylene glycol, which we've seen some reactions to in certain populations. Okay, so in the last few minutes, I wanted to go over the CDC recommendations, because if you go take a look at the CDC website, there's a bunch of charts like this, and they're all kind of somewhat confusing because you see all sorts of different colors, all sorts of different words. This is the vaccine schedule for children. And I'm just showing this. I'm guessing that most of you are not going to see this, although those of you with children will recognize this quite well. There's the vaccine schedule for children is very kind of regimented. It's set pretty strictly because we have vaccines we want to give at birth, hepatitis B, or within the first couple months of life. So rotavirus, DTaP, things like that, pneumococcal conjugates. These are all very well scheduled. And these, you go to the pediatrician's office, you see these guys at those particular developmental stages. Some of the other ones that are more for adults, you see COVID vaccines, some of the annual vaccinations, but also measles starts coming up after about 12 to 15 months. This is to improve vaccine take because of the efficacy of the measles vaccine. Most people that are properly vaccinated with measles have lots of antibodies, and those maternal antibodies get transferred to the infants, so they don't have really good vaccine take before 12 months of age. So you can see there's a pretty regimented within certain months. And then as you get into the teen years, it tends to be more of this annual vaccination that you see for the adults. If you take a look at the adult schedule, then it's a little bit different. The things that you can see are the sort of annual vaccines. So COVID-19 vaccines, flu vaccines, Tdap or TD boosters every 10 years. One of the things that you'll see here is this is varicella. So for those born in 1980 or later, you can get the live attenuated vaccine that's given to the pediatric population. There's a little bit of a gap, but once you're 50, you can get the shingles vaccine, the Shingrix, the recombinant protein vaccine. So varicella zoster vaccines are really still recommended throughout life. It's just the form of the vaccine that you get is a little bit different. Papillomaviruses are for young adults. It's mostly because it's to prevent sexual transmission. By the time you get to be 27 through 45, it's approved through 45, but by this time, if you haven't been vaccinated, it's more likely that you've already been infected. And then there's the pneumococcal vaccines, and these, it just says see notes, and I will go over this in a little bit because it is kind of confusing. So this is for healthy immunocompetent adults, but if you go to the schedules, then you have people with other indications that have a little bit of difference in which vaccines they should take. One of the things you'll see during pregnancy, no live vaccines. So that's contraindicated. The papillomavirus vaccine is also contraindicated during pregnancy. These live attenuated vaccines are also contraindicated for immunocompromised people and people with, progressing to AIDS. But again, you see with most conditions, you still, the COVID vaccines, flu vaccines, and then Tdap. So for pregnancy, Tdap is recommended during pregnancy, but again, this booster every 10 years. So there are differences in a lot of these types of the immunocompromised. For example, a lot of these killed vaccines or conjugate vaccines are recommended for people with HIV. Again, things like Mpox, these are not recommended for all adults, but recommended based on risk. So for Mpox, for example, men who have sex with men, that was the predominant population that was transmitting Mpox in this last outbreak. That would be the population that you would get. So a lot of these are actually a little bit confusing, but in general, people with some sort of medical indication, at least the killed vaccines or inactivated vaccines, subunit vaccines are recommended. Okay, to finish off, I just wanted to talk a little bit about some of the specific vaccines that I particularly find confusing. So the influenza virus vaccines, there are a whole bunch of different influenza virus vaccines. So for 2023-24, they're quadrivalent. There's the standard ones that are grown in eggs. These are the formalin inactivated vaccines. Most people can get these without problem. There is one that's made in cells. If people have, potential egg hypersensitivity, they can do this cell-derived flu cell vax or the recombinant form of the influenza virus vaccine called Flublok. This is just the protein itself. So these are denoted either IV. This is inactivated, cell-derived, recombinant. There's also a live attenuated flu virus, Flumist, that's only indicated for people from 2 to 49 years of age. For the elderly, there are 3 different types of vaccines. There's a high dose quadrivalent, which is basically just 4 times as much antigen as the normal egg-derived inactivated vaccine. There's the Flublok, which already has more antigen. That's also recommended. And then there's a quadrivalent inactivated vaccine that has adjuvant in it. So these are the ones that you are recommended for the elderly because of the immunosenescence that they see. The other thing that's going to be a little bit different for flu is this year it's quadrivalent. Next year, this coming fall, you're going to start seeing trivalent flu viruses. So one of the things that happened during the initial phases of the COVID pandemic is that we started doing a lot of non-pharmaceutical interventions that prevented spread of flu virus. So we had very weird flu seasons, but in addition to having weird flu seasons, we actually seem to have caused the extinction of this B Yamagata lineage of influenza virus. So that is no longer included in the vaccine. So when it says trivalent, now we're going back to a trivalent vaccine that has H1N1, H3N2, and then the B Victoria lineage. Pneumococcal vaccines. Pneumococcal vaccines, the recommendations are very complicated. And so there are really two different types of pneumococcal vaccines, the conjugate vaccines. So these are the polysaccharides conjugated to, I think it's diphtheria toxin, or yeah, I think it's diphtheria toxin they're conjugated to, as well as the polysaccharide vaccine. So the conjugate vaccines are approved for children and adults. Polysaccharide vaccine is only approved for specific populations above 50 and above 2-year-olds at risk of disease. The recommendations themselves are for the pediatrics. Again, it's really easy. Schedule 4 doses, 2, 4, 6, and sometime after a year of these 2 conjugate vaccines. For the elderly, so 65 and above, if you're not vaccinated with a conjugate vaccine or you've been vaccinated with the conjugate vaccine PCV7, then you have these particular recommendations, either just PCV20 or PCV15, followed by the polysaccharide vaccine a year later. And then there's some shared clinical decision-making for using PCV20 if you've had the other 2 vaccines, PCV13 or the polysaccharide PPSV23. These are all very confusing. If you really need to have the recommendations, they have a mobile app. CDC recognizes that their recommendations are really kind of confusing, so there's a mobile app for that. There's also a website on CDC that you can find, and I think Jennifer will drop this link into the chat as well. Okay, the last thing that's confusing are the COVID-19 vaccines. I looked at the recommendations, and the recommendations are kind of bonkers crazy. But the COVID vaccines, we have 3 that are currently in use in the United States. The mRNA vaccines are fully approved for individuals that are older than 6 months of age. So the Pfizer and the Moderna mRNA vaccines. The Novavax protein nanoparticle vaccine is still under emergency use authorization, but it's been authorized for individuals that are 12 years of age or older. Okay, so on the right here is the table that they put on the CDC website, and it has all these different things on it that are highly confusing to me. But basically, the idea is for the COVID-19 vaccines for kids up to 4 years of age. Again, they can only get the mRNA vaccines. If they're not vaccinated or they're partially vaccinated, you bring them to full vaccination status, which means 2 doses of the Moderna vaccine or 3 doses of the Pfizer vaccine. If they are fully vaccinated already, you can give them a single dose of the updated vaccine at least 8 weeks after the last dose. So this is the kind of updated flu. It's basically updating to the Omicron variants now that are present and circulating. So this is for 4 years and under, either bring them up to full vaccination or give them 1 dose of the current vaccine. For 5 to 11, it's easier. You basically just give them 1 dose of the updated formula of the COVID vaccine. Again, since they're below 12, this only means mRNA vaccines, and it has to be at least 8 weeks after the last dose. It doesn't matter where they're unvaccinated or vaccinated already, just 1 dose for the 5 to 11s with the mRNA vaccines. And then above 12, 1 dose of any vaccine if they are at least 8 weeks after the last dose. If they're vaccinated, if they're unvaccinated, for Novavax, if you're giving them that, you have to give them 2 doses to get them fully vaccinated with Novavax. However, if they're unvaccinated, you're giving them mRNA, you only give them 1 dose, like the 5 to 11-year-olds. So the basic idea is if you just use an mRNA vaccine, give 1 dose of the updated from anybody from 5 years of age and up. Okay, and then 65 is basically the same, 1 dose of the updated vaccine, or bring them up to full vaccination for the elderly. The last thing I want to touch on really is these RSV vaccines is really exciting for me as somebody who's worked on RSV vaccines for decades. We now have RSV vaccines. So a GSK version, a Pfizer version, they're both protein-based. They both use the same protein called RSV pre-F. And the difference is really the monovalent, bivalent, it doesn't really matter. The different subgroups of RSV are still similar enough that the immune response that you get is effective against both subgroups. The real difference between the two RSV vaccines is that the GSK has their proprietary adjuvant, this AS01, same adjuvant that you use in Shingrix, except at a half dose of the adjuvant. So if you have patients that have responded poorly to the Shingrix vaccine, this may be a consideration not to use the adjuvanted RSV vaccine. Pfizer does not have adjuvant in it. So these guys are both approved for above 60. And the Pfizer vaccine is also approved for maternal immunization between 32 and 36 weeks of gestational age. Before that is not as effective. After that age is not as effective. The reason that the GSK is not approved for maternal immunization is they had to stop their clinical trial because there was a signal that they had more stillbirths in their vaccinated group, and it's likely related to something that the adjuvant's doing in that particular group. So maternal immunization is only with Pfizer. What does pre-F mean? Pre-F is actually a really important development in the last 10 years, which made both the RSV vaccines possible, but also made the COVID-19 vaccines that we use more effective. What this means is that there's a fusion protein, there's a protein on the virus that allows for the virus to fuse with the cell that it's going to infect. This protein has a particular form on the outside of the virus called the pre-fusion form, waiting to contact the cell in order to trigger. Once it triggers, it has what's called a post-fusion, and that means it's already brung the viral membrane and the cell membrane together. So it looks very different structurally than the pre-fusion state. Turns out this post-fusion F is not, it's easier to make because it's energetically more stable as a protein, but this pre-fusion protein is really the one that you want to make because that's what's on the surface of the pathogen itself, and that's why you want your immune system to recognize. The post-fusion F vaccine actually failed in phase 3 trials. That was a Novavax vaccine. The reason for this difference is really that the post-fusion F doesn't have very many epitopes, and the epitopes that it does have are poorly the antibodies that you develop here are poorly neutralizing. The pre-fusion F has more epitopes, and the ones that they have tend to be highly to develop antibodies that are highly neutralizing, particularly these ones on kind of the head of the fusion protein and right here. So this turns out the site 0 turns out to be the site that you want to target in order to get neutralizing antibody. And just for reference, this is where this monoclonal antibody nirsevimab recognizes. I'll talk about that in a little bit. I'll talk about it right now. So nirsevimab then is this antibody that recognizes this top, and nirsevimab is also a new called RSV vaccine. So nirsevimab is actually a monoclonal antibody that has been modified to have a longer serum half-life, and it's been optimized for really tight binding to that site 0 of the RSV pre-F. It's given to neonates intramuscularly before 8 months of age as they enter their first RSV season. RSV season here in Florida usually starts in October. So babies that are entering their first RSV season can get this antibody. It's one shot, and it should protect over the entire season, as opposed to previously the palivizumab was required to be given every month during the course of the season. So at least 5 doses here in Florida. The one difference for nirsevimab, even though it's a monoclonal antibody, it's being regulated by the FDA as a vaccine. So it's going through the same vaccine regulatory processes that all of the other vaccines that we've talked about go through, rather than as an agent like the monoclonal antibody therapies that we use for a number of different immunologic and oncologic diseases. Okay, so now just to finish up, I think what I want to say is that really we have different stages of life that need different vaccine protocols. Maternal immunization has its own specific needs. You know adjuvant, generally protein-based, a non-replicating type of vaccine. In infants who have an immature immune system, perhaps the best idea is not really to try to protect them with vaccines, but maybe monoclonal antibodies would be best. And then children over 6 months of age, now you're starting to get into a place where their immune systems are developed, and you can protect the children with live attenuated or vector-based vaccines. And then as we get older and our immune systems start to age as well, you start thinking about different types of vaccines where you can have a more protein-based vaccine that will not cause as many, adverse effects as a live attenuated vaccine. And with that, I see my time is done. So thank you for being here, and thank you for your attention.
Dr. Ledford, did you want to ask your question that you had in chat?
Mike, thanks so much for that really important review, because it's something that as internists, we really need to be the champions, I think, for vaccination. But I guess my question was more of a philosophical one, and because you've had to deal with the press so much, your comments on how we deal with the fact that as the risk of disease declines, the relative risk of the vaccine increases, because people don't see the impact of the disease, and so any side effect becomes like magnified because they don't put it in the context of the suffering the disease causes. I don't know that there's any solution, but I was just asking if you had any clues on how we talk to patients to convince them of the value of vaccination.
Yeah, I mean, it is a real problem, and we're starting to see decreases in things like MMR, right? Vaccination rates here in the United States because people haven't seen measles virus, polio virus. I think with most of the vaccines, they tend to overemphasize these really pretty rare events. We're talking, even with the mRNA vaccines for COVID, we see myocarditis in young males, but the incidence is very low, and the disease is actually not nearly as significant as you would get if you did get, COVID itself. So I don't know exactly how to talk to people about it. I just emphasize that these are really, these are deadly diseases. That's one of the reasons, you can show the slide that I had in the beginning where hospitalizations and deaths from some of these diseases was pretty significant prior to vaccines being around. And now that just because we don't see the disease doesn't mean it's not there anymore. I don't know if that answered your question, Dennis, but
there is no answer. I just think it's something we have to struggle with, but be enthusiastic about vaccination, because as opposed to seatbelts, where it only affects the individual, people who choose not to vaccinate affects all of us.
right.
And it's just important for us to be the champion with nothing else, with just the inflection of your voice and the fact that you take it seriously. But thank you very much.
Totally. Thanks, Dennis. Dick, I see your hand up.
And one comment about that: they've taken the vaccinations out of doctors' offices, and doctors don't give vaccinations. And the best place to get counseling about vaccinations is from a doctor or healthcare professional. Now you have to go to CVS or Walgreens or Costco's or something to get a vaccine, and that's very much more impersonal without the input from doctors. It's a big mistake. And what happened is we try to give vaccines in practice, but they're so expensive versus what somebody like CVS has to pay when they buy 1,000 or 10,000, 100,000 vaccines at one time. They're so expensive, you can't make them cost effective to give. In my opinion, that's the big problem of vaccinations today. Pediatricians still have a pretty good chance of vaccinating people because they give the vaccines in their offices. Thank you, Mike. Very nice talk, and a very, very important subject to talk about. Thank you.
Thanks, Dick. Joe, your hands up.
Hi, yes. I was wondering, do you have any concerns about the messenger RNA vaccines being pushed, the other ones coming down the pipeline? Because first of all, we were given all these high hopes of them, for instance, protecting the individual, protecting the community, and we know that they've done a very poor job of that in the sense of obviously one can been vaccinated can get COVID again. It doesn't prevent transmission. And then we've had unprecedented levels of pharmacovigilance data all around the world, the yellow card system, the VAERS, the safety in terms of unprecedented numbers of adverse events associated with the vaccine. There's autopsy data. There is the evidence from studies done on the vaccine in terms of mechanism, the spike protein, for instance, being a toxin, contamination concerns. And then also, you can argue these are gene-based products, but we haven't done the genotoxicity studies. You can't turn off the messenger RNA once it's in the body. We were told that it would disintegrate very soon. First of all, it's not messenger RNA, it's modified messenger RNA. And so I'm concerned about the lack of oversight to really do the proper safety studies, and I think it's really increased the vaccine hesitancy, and I think that's part of the reasons why the public has been reticent to take other vaccines, because they've seen the dismal performance of the messenger RNA vaccines. And then to top all that off, you have the competing interest from the pharmaceutical company, which supplies 65% of the funding of the FDA coming from the pharmaceutical industry. So you have this conflict of interest as well. If there's not an independent agency doing the proper oversight and safety.
Yeah, I mean, I think these are all talking points that I've heard a lot before, and I think, frankly, just the numbers don't bear out your point. If you actually take a look, the mRNA vaccines, which were deployed mostly here in the United States, were pretty effective at knocking down the numbers. The high vaccination areas had less disease, less hospitalizations, less deaths than the lower areas of vaccination. Genotoxicity, it's an mRNA, it's a modified mRNA, modified by putting in naturally occurring modified nucleotides. So it's not something weird. It's something that's just there so that you don't turn on your innate immune response and prevent the mRNA from being translated. There's no, genotoxicity, it's an mRNA. It has no way to get to the nucleus in the first place. So that's just biochemically not really a thing. There's a lot there to unpack. I mean, yes, we do have incidences of one of the things that I do think is potentially a problem is I don't think we have the right lipid nanoparticle formulation for, because we do see some things like these myocarditis cases in young males. We don't understand necessarily why that is, but for sure that there should be improvements in the lipid nanoparticle formulation.
Mm-hmm.
Spike protein being being a toxin. I mean, if you're worried about the little bit of spike protein that's being made by the mRNA, you should be a lot worried by how much spike protein is being made by COVID infection. So, I mean, just to think that protein itself is a big problem compared to what you would get if you got infected is, this is, again, I think going back to Dennis's point that if you're worried about these types of things, instead of worrying about the big picture, which is severe respiratory illness from SARS-CoV-2 infection, then I think this is where we and the healthcare medical professions have to actually be a lot more careful about how we talk about these things, because focusing on the negative, and all the vaccines have some side effect. They're not all completely without side effect. Having that be the major thing rather than how well they actually work is, I think it's irresponsible for us as, healthcare providers, healthcare professionals.
It's controversial. It's not something we can resolve now, but I will follow you up with you on this, because there's a number of publications have come out questioning both the safety and the efficacy that I think
I know,
deserve
and
more
our
attention.
Surgeon General talks about it all the time, And, you know, the numbers really just don't bear in mind. He had to actually fudge the numbers in order to get some sort of problems with the myocarditis to show up. So he actually faked some of his data. This is a huge problem. I mean, there's a lot of people out there that are making money off of vaccine hesitancy.
But you
Even a
talk-
presidential candidate that is doing this. So I mean, it's a big problem. Amber,
I mean,
you have your hand.
Pfizer-
No, Joe.
it's been a-
Amber has her hand up. Amber.
Hey, Dr. Tang, great talk. Question for you. I heard that the Pneumovax 23 might be being phased out, like that.
Yeah.
I think it's Merck. They're not going to make it anymore.
Right.
Have you heard that? And I guess for us as immunologists, it's a little bit disappointing, I guess, because that is a true polysaccharide vaccine that we can use to assess response to that type of vaccine. But do you know anything about that?
Yeah, I think I heard that they're phasing out production, and I think, you know, based on the CDC recommendations, I think they're trying to phase it out too. So that may be, you know, both kind of working hand in hand where they're not going to make it anymore. I don't know how. Can you use the other vaccines to look at responses? The PCV, the conjugate vaccines?
Well, you can, but then you're looking at, B and T cell.
Yeah, you're
You're
also
very much
looking at
aware.
yeah.
So, I mean, it's more of an academic, I think, problem for us, and maybe like an ICD-10 diagnosis code problem. But
Right.
yeah, I just wondered if you had heard that also.
Yeah, no, they were, yeah, it looks like both the CDC and the company are kind of phasing it out because the conjugate vaccines work better.
Thanks.
Mm-hmm. Right. Yeah, look.
Oh.
Okay. So Jennifer's put the slide deck in a box folder, and that's linked. So you have access to all that information, whatever you need. All right, it's 1:10, so I will give you back your time. Thank you for
Thanks,
staying.
Mike.
For all you that
Thank you,
did.
Dr. Tang.
Yep.
Thank you.
Take care,
Thank you.
everybody.